Assalamualaikum warahmatullahi wabarakatuh.
My name is Lukman Hakim Ahmad Firdaus.
And my name is Asaidah Aisyah Faisal. We are from College Jenis Insan.
We are presenting school-scale Microbe Bioreactor kit introducing fermentation technology to the secondary schools.
Background study. The declining number of students interested in science, technology, engineering, and mathematics, based on the BED Asia Leadership Summit and Expo 2019 in Kuala Lumpur, poses worry towards the future of research and development in Malaysia. Coinciding with Chapter 6 of Form 5 Science subject related to food technology and manufacturing, the development of this knowledge was shifted to kit bioreactor innovations that apply microorganism fermentation technology in the production of food products such as cultural beverages, antibiotics, food preservatives, and even pharmaceutical products such as vaccine and alcohol. This innovation aims to increase the transfer of knowledge among secondary students in the field of biology and biotechnology by creating a school-scale microbial bioreactor kit ScMBK. ScMBK is a laboratory-scale bioreactor functioning with the fundamental parameters of industrial-scale bioreactors, such as airflow, impeller.
Introduction.
Fermentation technology is a field which involves the use of microorganisms and enzymes for production of compounds, which can be applied in the energy, pharmaceutical, chemical, and the food industry to produce vaccines, antibiotics, and food preservatives. Specifically, in biotechnology field, fermentation technology, mainly bioreactors, are used in the production of wine, vinegar, and cheese around the world. This conventional bioreactor has been proven for efficient production of bioactive compounds by applying submerged cultivation in the process, which offers potential advantage such as faster production in shorter time period within reduced space and lesser chance for contamination. Its importance also has been mentioned in Chapter 6 of Science, textbook Form 5 in Malaysia. In Malaysia, biotechnology is recognized as one of the economic engines that will drive the country towards a progressive and high-income nation. One of the key factors that contribute towards the success of biotechnology industry is the huge amount of highly qualified biotechnology graduates. Unfortunately, number of secondary school students enrolling in science stream has been declined. Based on statistics, only 19% of the approximately 447,000 candidates who sat for Form 3 assessment, which is known as PT3, entered the science stream when setting foot in Form 4. This concludes that educators' technique in teaching the student has not been quite appealing to interest the students. To date, there is no effective approach about the industrial fermentation technology to Malaysia's secondary school student and the invention of sophisticated prototype to apply a creative and interactive education in their learning. In this research, a prototype using only cheap everyday equipment was built to realize the form of homemade project or do-it-yourself DIY to simplify the delivery and assimilation of knowledge.
Methodology. The three main components for the ScMBK bioreactor will be the main body, air system, and impeller. For the installation of the main body, then 3 holes are penetrated using a drill to insert corresponding components such as the air inlet, air outlet, and impeller into the main body of the vessel. The holes should be aligned collinearly in a straight line. Now for impeller and power supply. The top of the metal straw is attached to a 2-voltage DC motor using a glue gun. Before drilling the hole for assembly of the L-shaped metal bracket, the impeller and L-shaped metal bracket are placed to their respective places. An outline of the hole where the nut and bolt will be placed is made. The L-shaped metal bracket is assembled. After assembly of the metal bracket, the impeller is inserted and tied to the metal bracket using a cable tie. After that, a battery holder with 2 batteries are also tied vertically to the metal bracket using a cable tie. After inserting the metal straw and the motor into the hole of the bioreactor lid, the bottom part of the metal straw should be attached approximately 6 to 8 cable ties These cable ties will now act as the propeller for our now constructed impeller. To continue, the air system. For the air system, a 50 cm and a 30 cm of silicon tube are prepared as the air inlet and air outlet respectively. The 50 cm silicon tube is attached to an air pump at one of its ends, while at the other end, a bubble stone is attached. By attaching the bubble stone to the end of the silicon tube, we are actually able to minimize the size of the air bubbles that is supplied by the air pump. Finally, after we have finished completing all of the three main components, we should assemble the bioreactor. Water is inserted into the main body and is filled until 3/4 of the whole jar. Then, the connecting wires should be attached to the DC motor so that the spinning mechanism begins. Then, we should also attach the mobile air pump into any power source using a USB cable so that it produces bubbles. Results and discussion. Here is the results of the bioreactor compared with the schematic diagram that we have prepared. This table shows each source, apparatus, and price per unit of the main components. For the ScMBK, it can be concluded that the total is 15 ringgit only, which is very affordable for secondary school students. The ScMBK is a relatively new research effort accompanying many school-scale kits currently in Malaysian secondary schools in endeavoring food biotechnology to be implemented in secondary schools. With the help of these projects, new ideas and talents can be made tangible by involving students with active learning instead of continuous input for examination purposes. The ScMBK uses a reusable large Nescafe jar, which is widely known and easy to find. It also requires apparatus, most likely to be found in department stores.
At the end of this project, targeted secondary school students can enhance and upgrade the caliber of science clubs in school. Educators can also use the ScMBK as a new perspective of learning and a crucial factor in education and can be made mainstream by contacting manufacturers for future projects regarding ScMBK. Conclusion. Based on the verse above, Allah has the absolute power of ordering all microorganisms to function beneath His command. All living organisms, including the ones minutely hidden from the naked eye, are not hidden from Allah.
Thus, it is important that topics in STEM, such as fermentation technology, be introduced to the Malaysian students to nurture new Islamic scholars in the future.